Okay, so now after detailed discussion on the detailed introduction of the Haemophilus influenza, now it is time to discuss about the virulence factor. So first of all, it is important to understand about what is virulence factor. So virulence factor is nothing but weapons of bacteria, okay, that allow bacteria to destroy the host cell and evade the immune system. Okay, so now let's see what are the weapons that the Haemophilus influenza possesses. So the first and major weapon is polysaccharide layer called capsule. Okay, because this capsule has antiphagocytic activity. Okay, and this antiphagocytic activity help bacteria to be protected from phagocytosis activity of macrophages and neutrophils. Okay, that means this antiphagocytic activity of the bacteria protects bacteria from the phagocytic activity of neutrophils and macrophages. Okay, and that's how bacteria can protect itself from the destruction and can attach to the epithelial cells in the airways. Okay, that means polysaccharide like capsules or polysaccharide layer called capsule help bacteria to attach to epithelial cell, right, by avoiding the destruction that is facilitated by macrophage and neutrophil via their phagocytic activity. Okay, and here capsule play a anti-phagocytic role. Okay, now how they attach? So there should be something for their attachment. Yes, obviously. So there is a pili. These pili are hair-like extension which help bacteria to attach to host cell. These pili are nothing but, let's say, this type of things. Okay, this type of things allow bacteria to attach with the host cell. Okay, and two extra adhesion protein present. These adhesion proteins are HMW1 and HMW2. Okay, so that is the idea of the virulence factor of polysaccharide layer, right? So polysaccharide layer, that means it is very important to understand that this virulence factor is only present in typeable Haemophilus influenza, right, or encapsulated Haemophilus influenza. So I would like to recommend you that if you have not watched my previous video regarding the introduction of Haemophilus influenza, then I would like to recommend you to go back and watch it first, because there I have explained the classification of Haemophilus influenza. Okay, so if you have you know about that Haemophilus influenza are of two types. One is encapsulated, another one is unencapsulated. Then it will be better for the understanding. Okay, so this virulence factor is the virulence factor for encapsulated bacteria. Okay, so it has antiphagocytic activity which protects the bacteria from macrophages and neutrophils. Okay, and allows the attachment with epithelial cells in the airways with the help of pili and adhesion protein. Okay, so pili here are nothing but hair like extension, and on the other hand adhesion proteins are HMW1 and HMW2, which facilitates the attachment of the bacteria with the host cell. Okay, so now after discussing about one virulence factor for encapsulated bacteria, now it is time to understand about one virulence factor for unencapsulated bacteria. Okay, although this virulence factor is present in both encapsulated and unencapsulated bacteria. So in previous video I have told you that Haemophilus influenza is nothing but a gram negative bacteria. Okay, and gram negative bacteria have a outer membrane. Okay, now in that outer membrane it have a special type of oligosaccharide known as LOS. Very important. So this LOS or lipo-oligosaccharide, it is a important virulent factor for both the capsulated bacteria and unencapsulated bacteria. Now let's see how LOS protect Haemophilus influenza. So this LOS will inhibit the mucociliary clearance, or it is the mechanism, that means I am talking about the mucociliary clearance. So mucociliary clearance is the mechanism of the self clearing for bronchi. Okay, so this self clearing mechanism of bronchi, or only the mucociliary clearance, help our respiratory tract to be bacteria free, right, by sweeping them out. Okay, so under such circumstances, loss inhibit this. That means mucociliary clearance or the self-clearing mechanism of the bronchi, and as a result, our Haemophilus influenza can happily colonize in the respiratory tract, right? So that is the idea. Again, so our loss that is present in the outer membrane of both the encapsulated and non-encapsulated strain, okay, have the ability to inhibit mucociliary clearance, which is nothing but the self-clearance mechanism of the bronchi. Okay, and as a result, respiratory tract cannot be bacteria free. Okay, so now as a result, due to the presence of loss, Haemophilus influenza can easily colonize. Okay, another important virulence factor, that is the Iga protease. Okay, so remember it very clearly that in the nasopharyngeal mucosa secretions there is a special type of immune protein called Iga, that is immunoglobulin A. Okay, and it is a secretory protein and it is a dimer. Okay, mind it very carefully. So this Iga will opsonize the bacteria. Okay, and opsonize, what is the process of opsonization? So opsonization is nothing but it helps the neutrophils. I hope you know that neutrophils is a immune cell. So this immunoglobulin A helps neutrophils to recognize the bacteria. Okay, and as a result, destruction of the bacteria facilitates. Okay, that means the job of Iga is to facilitate the destruction of the bacteria by neutrophil, okay, via a process called opsonization. Okay, so now this Iga protease, as it is a protein breaking enzyme, so it will break the Iga protein, and as a result, the first defense mechanism of the mucosal cell become disrupted, okay, or neutralized. So encapsulated or an unencapsulated strain have two major virulence factors. One is the loss, that is lipooligosaccharide. This lipooligosaccharide inhibits mucociliary clearance, and also the Iga protease. This Iga protease destroys the immunoglobulin A, okay, and neutralizes the first defense mechanism of the mucosa. Okay, so that are the two most important type of the virulence factor. One is the polysaccharide layer called capsule, which is only for encapsulated bacteria. Okay, this polysaccharide layer capsule have antiphagocytic activity, and it protecting bacteria from phagocytosis by macrophages and neutrophils. Okay, and as a result, bacteria now can get the freedom to attach to the epithelial cell of the airways. Okay, and this attachment is provided by another two important part of the capsule known as pili and adhesion protein. Okay, an example of adhesion proteins are HMW1 and HMW2, and we have also discussed about two other virulence factor which is present in both the encapsulated and unencapsulated strain. One is the Iga protease, which destroys the Iga present in the nasopharyngeal mucosa secretion, okay, which causes the destruction of the Iga, and as a result, the first line of defense mechanism become neutralized. Okay, and another important thing, as it is a outer membrane, there is a lipooligosaccharide present on the outer membrane. Okay, and this lipooligosaccharide is nothing but it inhibiting or it deactivating the mucosal clearance, okay, or mucociliary clearance, which is the self-clearing mechanism of the bronchi. Okay, and as a result, respiratory tract cannot be bacteria free because this self-clearing mechanism of the bronchi so in encapsulated and unencapsulated strain we are talking about loss, right? So this loss causes the mucociliary clearance inhibitor, right? And due to this, the respiratory tract cannot be bacteria free. Okay, so that are the various kind of virulence factor. So in first we have discussed about the polysaccharide layer called a capsule. Okay, this capsule have phagocytic activity, right? Not phagocytic activity, obviously it have antiphagocytic activity, right? So antiphagocytic activity means it protects the bacteria from phagocytosis caused by macrophage and neutrophil. Okay, and as a result bacteria can get the chance to attach to the epithelial cells of the airways. Okay, and another important thing, this attachment is provided by pili and adhesion proteins, that is HMW1 and HMW2. And encapsulated and unencapsulated strain have an outer membrane extra, which outer membrane consists of loss, and this loss have the ability to inhibit mucociliary clearance and also the self clearance mechanism of the bronchi, which involves in the sweeping of bacteria out of the respiratory tract. And as it gets inhibited by the loss, now our Haemophilus influenza can easily colonize the respiratory tract. Okay, and also Iga Protease is there that utilizes the first line defense of the mucosal layer. Okay, now it is important to discuss about how unencapsulated strain evade the defense mechanism, right? Because as we have studied that encapsulated strain have polysaccharide, right? Polysaccharide like layer called capsule, right? So this capsule acting as an antiphagocytic agent and protect bacteria from phagocytosis. It is quite clear, right? But as compared to encapsulated bacteria, uncapsulated strain are less protected because it lacks that antiphagocytic part. Okay, that means if macrophage and neutrophil can somehow recognize it, it will immediately causing the phagocytosis, right? There is no doubt about this. So how unencapsulated bacteria causing infection? Because in the first video we have discussed that unencapsulated or non-typable bacteria are also involved in disease causing part. So how they are doing this? So this is now our topic of discussion. So they simply evade the immune system with the help of two different mechanisms. Okay, so one is phase variation, we will going to discuss, and another one is the biofilm formation. So now it is important to understand about what is phase variation. So remember that we have told that there is a oligosaccharide present in the outer membrane, right? So in case of unencapsulated strain, those oligosaccharide can change its makeup. Okay, each time it is infecting a cell. Okay, and as a result, for the immune system, it becomes very hard to detect detect and remember about the infecting strain. And as a result, whenever that same strain again come to infect the body cannot generate a specific and immediate immune response, because the oligosaccharide which is acting here as an antigenic agent, obviously this oligosaccharide changes its makeup each time when it infecting a cell. Okay, and as a result the immune system of our host defense cannot remember about them. Okay, and as a result the immediate and specific immune response, which is a characteristics of the secondary immune response, cannot be generated here. Okay, that is the most important feature of the unencapsulated strain, you know. And another one is unencapsulated strain have the ability to produce a layer of group like substances called biofilm. Okay, so these biofilm are made up of exopolysaccharide, that is EPS, very important for MCQ. Okay, so in the biofilm we have Haemophilus influenza. Inside is it, those Haemophilus influenza live and reproduce. Okay, and they remain away from the any kind of phagocytic activity, any kind of immune system action, and any kind of antibiotics that we are providing from the outside. Okay, so that is the two defense mechanism that unencapsulated strain have for their own protection. One is the phase variation, where it changing its oligosaccharide constantly whenever it is infecting a cell, and as a result it becomes very hard to remember for the immune system about the infecting strain. Okay, as a result secondary immune response cannot be generated. And the second one is biofilm, which protects the bacteria from antibiotic attack and other immune system attack. Okay, and here we have also discussed about the virulence factor. So the first and most important virulence factor is the polysaccharide layer called the capsule. This capsule have antiphagocytic activity and protects the bacteria from phagocytosis and allow them to attach to the epithelial cells of the airways. So this attachment is done with the help of pili and also to adhesion molecule called HMW1 and HMW2, and obviously there are presence of Iga protease which destroys the Iga, and also the loss, that is our lipooligosaccharide. Okay, this lipooligosaccharide is doing what? This lipooligosaccharide is inhibiting the mucociliary clearance. Okay, and as a result the bacteria can colonize the respiratory tract. Okay, so this is all about the virulence factor of Haemophilus influenza. So I hope this video will be helpful to you to understand the concept of the virulence factor. So we will again come back with another video lecture where I will explain which kind of bacteria causes more invasive infection and which one not. Okay, so till then, bye bye.